What you're looking at right now is an example of a genetically modified E. coli cell that I made almost exclusively using Claude Code. As you can clearly see, it's been engineered to both glow fluorescently green, but it's also been engineered to resist the drug ampicillin. But these are just two of a variety of edits that you could possibly make in this mini genome editing lab that I've created all inside of Claude Code. So in this video, I'm going to break down exactly how I built it, what information it uses to work successfully, and how, again, it's such a super simple process to build. So let's get into it. So the way I'm going to be building out this web app is by a prompt basis. Essentially, I'm going to do first main prompt, second main prompt, and then I'm going to use a prompt to initiate the kind of sequencing genome side to it. This is just so the whole thing doesn't confuse Claude all at once. If I give it all the information, it might hallucinate, it might do tasks less efficiently as if there was just one given to it at a time. So the first prompt, as you can see right here, revolves around nailing the aesthetic. Now, the aesthetic I wanted to have is kind of a video game action adventure menu style aesthetic. That's just the idea I have in my head. Of course, you can only choose anything for that, but I think this will suit the kind of theme that we're going for here in this laboratory environment. Another really important thing to note here, though, is that Fable 5 safeguards will flag any message that is biologically related, no matter how insignificant it may be. So in this first prompt, I've really made it specific not to mention any aspect of the web app being biologically related. Therefore, I'm able to use Fable 5 and its amazing design capabilities to design the actual web app first, and then we can input the data using Opus, because it's just not going to allow us to use Fable for that part of the process. Okay, so let's go ahead and give it over to Claude and see what it's going to come back with. Okay, nice. So it seemed to have done a really good job. Again, it's kind of nailed that video game menu type aesthetic, which is exactly what I was going for. Information doesn't mean much, but that's fine. We didn't ask for that. But yeah, looks really nice, and it's the aesthetic I wanted. So I'm pretty happy with that. So the next part of the process is going to go ahead and give it that second prompt to add the information to it that's going to actually make it functional. Okay, so let's go ahead and give it this second prompt. Now, the second prompt is a lot more complex. It's not just asking for aesthetic. It's asking for Claude to input information related from good sources into the app. As you can see here in the second prompt, what we're essentially going to be asking it to do is to create an interactive genome map, but also a semi-interactive organism page where we can actually view a diagram or a visual representation of what the organism looks like under those specific genome edits. So it's a bit more visual. So this website, E.Coli, is going to allow us to obtain all the information necessary to build out this genomic map for E.Coli. It's going to give us relevant and true data for Claude to use. At the start, we're going to add a few editable genes, so the likes of GFP, BLA, related to that drug resistance I mentioned at the start. Again, we're going to keep the amount of genes low so that things don't get too complicated. You could add as many or as little genes as you want, but I think it's best to focus on a select few so we can modify it in select ways. All right, nice. So we've gone ahead and given that to Claude, and it seems to have come back with something that looks really, really good. In terms of an informational point of view, of course, it's the same aesthetic, but we can see here that we have multiple new tabs. And I think it is a great process in actually creating something that's informationally relevant. One thing I think we can improve on lots, though, is the actual circular genome that it put into the web app in the genome tab. Now, as you can see here, we can inspect each locus. It looks nice. It is okay, but I know we can do a lot better. So for something like this, what I typically do is I would take inspiration from something like Pinterest. You can search images as well, but Pinterest is just such a great source for this kind of thing. If I was to go open source circular genome, it would give me a whole bunch of designs, whatnot, that I could give to Claude so it could kind of reimagine how it's going to create the circular genome diagram. But even the organism tab, although basic for now, it still functions relatively well. But I will say the circular genome, I think, can be much improved on. So I think we should go ahead and improve it. As I said, if we just hop over to Pinterest here and even taking a look at some of these images, I mean, they look far better. Obviously a lot more complex, but we can always dumb down the complexity a bit in Claude and focus on the genes we only really want to see. So I'm going to go ahead and tell it to rework the genome map. Again, it may be designed, but I'm going to have to switch back to Opus simply because Fable won't let me design on its terms. Okay, so I think it looks good. I don't think it looks fully there yet. I'd slightly enlarge it and maybe add a few more genes so it just looks a bit more populated. We don't even have to make them editable genes though. Okay, yeah, this is a bit more along the lines I'm going for. Again, we can see a much smaller proportion of it at any one time, but it just looks a lot better. Even though I've added more genes and more clickable genes for that matter, it's important to note that we will keep the amount of editable genes small simply because we want them to be detailed and we want them to actually reflect the changes in the organismal point of view. Okay, nice. We can even inspect each locus and see what genes around it. So it looks good. It functions well. So I think we'll take it from there. Okay, so the next part of the process is actually adding this genome splicing page so we can actually go ahead and make edits to the organism. Now I thought about this and I used to want it to be on the genome page. So I wanted to be able to click on certain areas of the genome and kind of drag and drop, taking bits in and putting bits out. But I'm kind of realizing that that is going to be a little too complex. I don't even know where parts are going to go and I'm trying to make this. So it's just not going to be practical for this. I think it's a little too difficult. So what I'm going to do instead is going to try and get it to make a separate page where we can make edits to the organism by just toggling genes on or off. Nice. This seemed to come out pretty well. Okay, nice. It's exactly what I wanted there. Keep it simple. If we have a set number of genes, we can turn whatever we want on and whatever we want off, splicing in and out. And we check it out here. Yeah, so it reflects it very nicely in the organismal point of view as well. I'd still probably get rid of these guys on the right hand side. They're probably not as useful, but for now it doesn't really matter. Good layout. Yeah, good amount of genes. We can change structure. We can change, metabolism, regulation. Okay, nice. Looks good. Okay, so the next step of the process is again something more visually related, but I think it'll just take this web app a step above. And that's going to be using Blender via Claude to add in animated structures of the proteins that these genes that we can splice in or out will encode for. So here's my prompt. Again, very simple. We're just going to do a prompt number 1 where we're going to get Claude to structure a frame of a protein instead of the actual animation. Once that frame looks good, which it most likely will, we'll be able to move on to the animated side. Now to use Blender via Claude, it's super simple. All you're going to do is just tell Claude to use Blender to download it. To animate these proteins, you're going to need 2 things to point Claude at. Tell it to get the molecular nodes add-on. That's really important as well. But also to use something like AlphaFold, which is essentially a huge library of hundreds of thousands, if not millions, of different proteins and their structure, which is originally decoded by AI systems themselves. So using something like AlphaFold, Claude is almost guaranteed not to get it wrong. And we can see that come to life in our first frame here. This is of GFP, green fluorescent protein, and it's done absolutely perfectly. So all we're going to tell Claude to go and do now is to animate this. We're only going to want to do select few instead of it going through and animating the whole thing because that'll take a couple of hours. So we do 4 genes that we can take out, 4 genes that we can parse in. Okay, so nice. Claude has gone and done GFP animated and it seems to look really nice. This one as well. Yeah, perfect. Again, not hugely necessary in terms of the information point of view, but it allows us to see the protein that the gene codes for. Again, just making things a bit nicer visually. Just my opinion here. And if we check on AlphaFold here, which is the website we're using, we can see again, GFP exactly similar to what Blender has cooked up or Claude has cooked up for us. So again, super simple to do this. Yeah, splicing in, DNA, K. Yeah, looks really good. Okay. Yeah, all looks good. Okay, so the last step in actually cementing the aesthetic of this web app is going to be making a much better diagram of the organism in this session, which is going to be E. coli. The one Claude has is a little too basic. It demonstrates the features, the changes, I should say, that we can enact, but it's just not the best for this circumstance. So I'm going to use Higgs field at the MCP, which you can access super simply. You just want to hop over to higgsfield.ai, and somewhere on the homepage, you're going to find it around, here. You just want to click on that, and again, it just gives you the instructions to set up the MCP inside of Claude. Super simple. So once your MCP is set up, the Higgsfield MCP, I find it best to give Claude a couple of reference images. In this case, I'm going to be giving it an image of a bacteriophage, which is a virus, but it just looks hyper realistic, again, from Pinterest, and a actual bacterial cell, so it knows what it's designing. Once I've given it both of these, you just want to tell it to use the Higgsfield MCP and animate a E. coli cell. Again, very simple prompt, and it should come back with something that looks really good visually. And what did I tell you? It's really good. Far, far better than what Claude could do on its own, obviously. It's using frontier image models, so it looks a lot better. Here we can see our lovely diagram. Again, functions perfectly, looks really, really good, hyper realistic. I also should add that I've added in this test survivability here, which I'll show off in a second. If I just make some edits, let's see. Okay, let's turn off the flagella, turn on GFP. Okay, yeah, perfect. Very clearly you can see it glows green, has no flagella or tail kind of at the end of the cell. If we click on test survivability, you see, again, non-viable. Now, the reason I've added this in is to kind of add another information layer for people using this. Again, it just shows, is this mutation or is this change going to affect it negatively, positively, neutral, whatever. As we can see very clearly here. Okay, so probably had that in before, but the mutation removing the flagella or tail was neutral. You can see GFP is neutral, it's just a color change. But we have something that altered the kind of the cell membrane that was negative. So that's why it was 24. Now if we try and toggle some other ones, we can see if we toggle that off, again, it's neutral 92% because the other ones are obviously neutral. We try some more. Again, there's so many combinations you could possibly make here. Let me turn on this antibiotic resistance gene, pretty sure. Again, just has the nice animation for the resistance. And that's going to be a more positive mutation or change that affected the genome. So you can see there the positive change. So there you kind of have it. In essence, this is what this app is all about. There's many, many changes you could possibly make. Obviously, you could dive into selecting specific genes, putting them in, the web app, obviously if they're not there, and then making those changes, seeing if they're positive or negative. But yeah, as I said at the start, this is super simple, super easy to set up. Mostly about the visuals, being able to actually visualize the changes made to the cell is really helpful and just pretty cool. But it also maybe in more practical sense lets you understand what the genes do and whether they have a positive or negative effect overall. So in totality, that was the super simple process of setting up this genomic editing lab for just one organism. Obviously, you could do many organisms. There's many changes I would make to it to strengthen it, to allow new capabilities. But please do let me know what changes you would make to it and maybe what similar projects you've created yourself using Claude Code. Honestly, the opportunity here is endless with this type of stuff. This is really only scratching the surface. But anyway, thank you for watching. See you in the next one.